Hello, everyone. This is a presentation by me, Kaylee, as well as my partners, Brittany, Amalia, and Kaitlyn. Our disease of choice was the bubonic plague. So what do you think about when you hear the bubonic plague? For us, before we started our research, it was rats, and you'll find out why that is later on in the presentation. But just to get your mind rolling, what do you think about when you hear the bubonic plague? So first, I'm going to get into a little bit of background on the bubonic plague. This disease ravaged Asia and Europe during the 14th century. It caused the most fatal pandemic in history, which is a really crazy statistic. There is genetic evidence of this disease from around 1348 that was found in plague burial grounds in Asia. The bubonic plague is believed to have come from a ship coming from Asia to Europe, carrying sailors and accidentally carrying rats that were likely infected by this disease. I'm going to get into a death toll statistic here that is really crazy. So the bubonic plague is estimated to have killed around 75 to 200 million people. And you might be wondering, why is this number so variable? And that's because the bubonic plague has been around for so long, as we mentioned, around the 14th century. So the exact number of deaths is still unsure because it has been around for so long, and it might have been around before that. And we researchers didn't know yet. And it is still around today. So the death toll continues to rise, and that's why this number is so variable. But to give you a little bit of a platform, around the time that the bubonic plague caused that first true pandemic, it is estimated to have killed around 50 million people. So to give you a little bit of a real world comparison, the COVID-19 pandemic, in which we are experiencing right now, as of a couple days ago, has killed around 4,700,000 people, which is a lot. But when you compare it to this death toll seen by the bubonic plague, it doesn't seem that big. So that is just an interesting thought. Here's an overview of what our presentation will look like. First, we're going to get into the agent that causes the disease, as well as the characteristics of the microorganism that causes the bubonic plague. We're also going to talk about the mode of transmission. So how is it passed from individual to individual? We're going to describe the characteristics of the bubonic plague, as well as how it is diagnosed, and is there a treatment? If so, what is it? Lastly, we're going to provide you with an interesting fact about the bubonic plague that you might not know. So let's get right into it. I'm first going to talk about what causes the bubonic plague. The bubonic plague is caused by a bacteria called Yersinia pestis. Yersinia pestis is present as a gram-negative coccobacilli. And coccobacilli basically describes a shape that is a very short rod or an oval shape. You can see in the picture to the right that this bacteria is presenting as gram-negative. And we know that because in this gram-stained picture, the bacterial rods are seen as a pink or red color, which is characteristic of gram-negative. When grown in culture, they are seen as a gray-white color with translucent colonies. They grow the best at 25 to 27 degrees Celsius, which is room temperature. And they can also grow around 35 to 37 degrees Celsius, which is a little warmer. So they have some variability in the temperatures that they're able to grow at, which makes it increasingly dangerous. However, they are easily destroyed by sunlight and drying. So in order to grow them, they need to be kept out of sunlight and they need to be kept in a moist environment. When a negative stain is attempted, the capsules are present on Yersinia pestis. So like we did in lab a couple of days ago, when we do a negative stain, we will see capsules in Yersinia pestis. Finally, where are they found naturally? This bacteria is found naturally in many wild animals, as well as squirrels, rabbits, and rats. So if you think back to my beautiful picture of the rat on the very first slide. Yersinia pestis is found naturally in rats.
So exactly how is the disease acquired? Or when we talk about mode of transmission, we talk about that first, the bacterial inoculation occurs from the host. When referring to bubonic plague, this is typically via the bite of a flea vector that acquired Yersinia pestis from primarily rodent species. The bacteria then travels from the dermis via the lymphatic vessel and into the draining lymph nodes. Majority of research indicates that the non-modal bacteria travels via a phagocytic host early on in the disease process. However, recent studies indicate that it may in fact flow with lymph fluid. The modes of dissemination is really still a focus of study in regard to this bacterium. So they're not all in all quite too sure 100% of how it travels from beginning to end. So though commonly bubonic plague is more prevalent in the focus of our presentation, it is important to know that if Yersinia pestis enters the bloodstream, that septicemic plague can result. And if it enters the lungs, pneumonic plague can result. When pneumonic plague results, the mode of transmission can then occur through production of airborne droplets. So this is just a slide showing the environmental transmission as well as the interhuman transmission. So through environmental transmission, aerosols can cause spread of the disease, as well as oral contamination, transcutaneous contact, which is really through like a bite or penetration through the skin, and then through wild fleas. Interhuman transmission can occur through aerosols, which commonly happens with pneumonic plague, and also through various flea species. So how exactly does Yersinia pestis invade the host defenses? So this bacterium uses a variety of mechanisms to evade or overcome host immune system, especially the innate immune system. It overall reduces the host adaptive immunity by both influencing the cytokine induction and acting directly on the immune cells involved in the adaptive immune response. So though we're going to cover some major mechanisms, this is not an exhaustive list. So in order to survive and or grow in the bloodstream for transmission between its flea vector and the mammalian host, this bacterium has developed resistance to complement-mediated lysis or serum resistance. So ultimately, it needs to be able to have adaptations to survive in a variety of hosts. Furthermore, they infect macrophages possibly via recognition of specific surface-associated CCR5 molecules to survive inside the macrophages and travel throughout the various portions of the body. The plasmid's PCD1 is really important to talk about because it's the most studied genetic determinant of Yersinia pestis. It's truly essential for infection. The PCD1 encodes proteins of the Yersinia type 3 secretion system. This is a syringe-like molecular apparatus responsible for secretion of proteins that interfere with host cellular functions. And then lastly, YOPs. YOPs produce plasmid encoded Yersinia outer proteins. These are necessary to downregulate antibacterial responses that constrict bacterial survival in the host. So to really summarize, what's happening is that Yersinia pestis has adaptations and abilities to truly lower our defenses and to almost paralyze or inhibit any type of antibacterial response to the disease. So let's talk about environmental factors. Species richness is a positively correlated factor with the presence of human plague. So an increase in rodent species increases the presence of the human plague. Furthermore, precipitation levels affect plague transmission by influencing the rodent and flea population densities, which in turn affect the spillover chance of plague into human populations. When plague outbreaks occur in larger human populations, they in general result in larger outbreaks of the disease as well. either directly in the case of human to human transmission or indirectly in the case of rodent-borne plague outbreaks. And lastly, transportation routes can affect the presence and intensity of outbreaks. It is partially correlated to human population density and can reshape the distribution of plague by accelerating the spread of infected animals and humans in larger areas.
Bubonic plague is the most common plague, but infection may also bring about pneumonic or septicemic plague based on what part of the body is involved. Pneumonic plague is the rarest, but the most dangerous because it can spread from person to person via cough droplets. In bubonic plague, the incubation period is usually 2 to 5 days, but can vary from a few hours to 12 days. Septicemic plague may occur with the bubonic form or without the bubonic form, which would then be called primary septicemic plague as an acute severe illness. Primary pneumonic plague has a 2 to 3 day incubation period. The mortality rate for untreated patients with bubonic plague is about 60%, but most deaths result from septicemia in 3 to 5 days. Most untreated patients with pneumonic plague die within 48 hours of symptom onset. Septicemic plague may be fatal before bubonic or pulmonary manifestations predominate. As a warning, the next few slides do include some pretty graphic photos as they are symptoms. The first symptom is swollen lymph nodes called buboes that typically develop in the first week after you become infected. Buboes may be situated in the groin, armpit, or neck. They're about the size of a chicken egg and will be tender and firm to the touch. Other bubonic plague signs and symptoms may include sudden onset of fever and chills, headache, fatigue or malaise, and muscle aches. Septicemic plague occurs when plague bacteria multiply in your bloodstream. Signs and symptoms include those of bubonic plague with the addition of abdominal pain, diarrhea and vomiting, shock, the blackening and death of tissue called gangrene in your extremities, most commonly the fingers, toes, and nose, as well as bleeding from the mouth, nose, or rectum, or under your skin, commonly called hemorrhage. Pneumonic plague affects the lungs. Signs and symptoms can begin within a few hours after infection and may include cough with bloody mucus, difficulty breathing, nausea and vomiting, and chest pain. Plague is a consideration for people who have traveled to the Western United States or another plague endemic area. The clinical diagnosis of plague depends on identifying the causative bacteria in fluid or tissue samples. The most common symptom is development of a bubo, seen here at the inguinal lymph node in the groin. The diagnosis is confirmed by staining, cultures, and serologic and PCR testing. Rapid diagnosis of plague is important because mortality increases significantly the longer treatment is delayed. Mortality is anywhere between 30 and 100%. Diagnosis is ultimately made by stain and culture of the organism, typically by needle aspiration of a bubo, as surgical drainage may disseminate the organism. Blood and sputum cultures should also be obtained. Other tests include immunofluorescent staining and serology. A titer of over 1 to 16 or 4-fold rise between acute and convalescent titers would be positive. PCR testing, if it's available, is used as a diagnostic tool. Patients with pulmonary symptoms or signs should have a chest X-ray, which would show a rapidly progressing pneumonia if you have pneumonic plague. The white blood cell count is usually 10,000 to 20,000 MCL, with numerous immature neutrophils.
Bubonic plague without treatment will likely result in death. It can cause infection throughout the body and become 2 other types of plague, septicemic and pneumonic. Treatment of the plague involves a number of multiple antibiotics: ciprofloxacin, levofloxacin, moxifloxacin, gentamicin, and doxycycline. For more serious cases of infection, oxygen, intravenous fluids, and respiratory support is given. The downside to treatment is antibiotic resistance. The long-term usage of antibiotics could cause the body to build up a resistance, thus decreasing the efficiency for future antibiotic use, and also organ damage. Longer-term treatment can damage internal organs such as your immune system and your kidneys. In regards to a vaccine, there was a killed whole cell plague vaccine that was used in the past, but recent studies in animals have shown that this vaccine offers poor protection against pneumonic disease. And there is a live detuned vaccine that's also available
It is effective, but it isn't considered suitable for humans, and it's not offered in the US because of that. So research now is research on Yersinia pseudotuberculosis as a live vaccine. It shares genetic identity with Yersinia pestis, and it's deliverable orally. However, it poorly protects against pneumonic plague, which is why that vaccine needs to be researched more. And currently there's research on an encapsulated version of that, which would better protect against pneumonic plague, so it would be more efficient. So an interesting fact that we found is though the bubonic plague is associated with Europe and Asian pandemics from many, many years ago, it is in today's world, it's still present around the world and prevalent. So Madagascar in recent years suffered outbreaks leading to fatality of hundreds. To the right is a distribution of plague cases and case fatality ratio by year of reporting from 2010 to 2017. So you can see that it is still prevalent in cases. However, we have developed methods to truly treating it before it leads to fatality in most cases. China alone had reported several outbreaks just last year in the midst of COVID even, and then the US averages about 7 cases a year. So these are our resources if you have any interest in diving further, and we hope you enjoyed our presentation. Thank you.